Yeah, so I'll give a very brief introduction to Jackson because I think he's going to do a good job of introducing himself as well. But Jackson's a second year PhD student in our lab. He comes down from the University of Washington, where he was also in the bioengineering program there, and has really quickly been putting together a data integration effort, looking at infection response and how we can find patterns between patients that resolve their infection versus don't. So Jackson, you can take it away.
Great. Yeah. Thanks, Dr. Meyer, for the introduction, and thanks for the QCBio for organizing this and having me here this afternoon. Let me go ahead and share my slide deck. Great. Is that visible?
Yep. Perfect.
Awesome. Oops. Let me get slides. All right, good afternoon, everyone. Today I'm really excited to talk about some of our work in applying tensor factorization for interpreting the mechanisms of MRSA persistence. But before we do that, just a quick introduction about myself. As Dr. Meyer mentioned, I graduated from the University of Washington with a Bachelor's of Science in Bioengineering. There I studied a lot of things, primarily type 1 diabetes, and I also got involved in a few global health efforts. So over here at the top right is this global health effort I co-founded called Insulin Anywhere that's looking to help improve insulin accessibility in these sorts of low resource and disaster response scenarios. Between then and now, I also took a gap year at Pacific Northwest National Laboratory, where I also do a little bit of consulting for to this day, working on a variety of projects ranging from things like studying type 1 diabetes progression to understanding sensed information and its sort of behaviors and errors. And so it kind of leads to a sort of interesting intersection of research interests today, where I'm really interested in things like infectious diseases, host pathogen interactions, and also sensed information behaviors. Which usually begs the question on how do all those things, how are they all related? How do I justify doing all that at once? And my answer that I give is that our data is never ideal. And it's usually a proxy for something more difficult to measure that we're usually more interested in. So both sensed information and biological data have this very unique problem where data is always kind of noisy, always kind of prone to bias and error. But the questions that we want to answer, they are in that data somewhere. And it's more about how we interpret that data to answer those more difficult and interesting questions. Other than that, I also have a dog that didn't fit on those previous slides. This is Kiki. She's my 7-year-old Samoyed, and she loves her outfits. So just a quick slide about her too. So to the actual research itself, the problem that we're addressing here is methicillin-resistant Staphylococcus aureus, or MRSA. And what MRSA is this increasingly common antibiotic-resistant strain of bacteria. The CDC estimates that about 2% of Americans carry MRSA somewhere on their bodies, though it's usually commensal. So it doesn't typically form these sorts of harmful infections. But in these sorts of rare cases, it can form really severe and even life-threatening infections. As the name implies, MRSA itself is also really difficult to treat. It's resistant to a lot of our favorite antibiotics, including treatments like penicillin. So usually in these milder cases, we rely on simply draining out the infection site and hoping that the immune system is able to clear it out. But in more severe cases, there is this really small subset of antibiotics that MRSA has shown susceptibility to that we can use in case the infection becomes too severe or potentially life-threatening. This includes treatments like vancomycin in particular, which I think is the standard use in the United States. The problem with these antibiotics, though, is that even if we know MRSA is susceptible to them, they often fail to clear an infection. And I think that's been illustrated by this flowchart here, where we can start with some patient with a MRSA infection. And from that patient, we can derive a portion of that infection and find that in vitro, this medication will clear that infection, will effectively kill off this MRSA. But what we find is that when we give that same treatment to the same patient, that in vivo, it fails to clear this infection. This is an issue we call persistence. And more specifically, it's when a infection shows susceptibility to a treatment in vitro or in a lab setting, but fails to clear an infection in vivo, or when we actually give it to a patient. And for MRSA, this is a really big issue, and that 30 to 50% of all MRSA patients become persistent, or their infections will show this sort of antibiotic resistance. And what's more, this persistence leads to worse clinical outcomes and universally more severe infections. So we find that patients with these persistent infections are more likely to have metastatic infections. So the MRSA is more likely to enter the bloodstream and migrate to another part of the body. They're more likely to have longer hospital stays. They're less likely to be cured, and they're more likely to die from the infection, which overall points to MRSA persistence leading to significantly worse clinical outcomes and much higher disease severity. And so a lot of research has been focused on trying to better understand how MRSA persistence happens. And one of the common theories in this area is that since there's a difference in vitro and in vivo outcomes, that the mechanism of MRSA persistence likely have something to do with the human body, since that's the main difference between in vitro and in vivo settings. And so there's been a lot of research looking into how MRSA interacts with the body and trying to identify these sort of determinants of MRSA persistence. One of the challenges in this area, though, is that we find that these determinants manifest across a variety of different biological sources. So even at like a simple genetic level, we find that there are genetic predispositions and immunodeficiencies that can make an individual more susceptible to both MRSA and MRSA persistence. At a gene expression level in the RNA space, we know that MRSA has the ability to modify methylation and can modify gene expression in ways to favor bacterial growth and to suppress the immune system. And finally, even from a protein level, MRSA infections have been associated with upregulating things like certain cytokines that are known to be anti-inflammatory and immunosuppressive as a way to suppress the immune response to these types of infections and to prevent clearance. And so what we're finding again is that MRSA persistence really is, it does involve the host immune system, or it does involve the host response itself, and that these mechanisms reflect across all these different modalities. But one of the challenges we find with each of these individual determinants is that none of them seem to be predictive. No single DNA marker or RNA signal or protein signal is usually predictive of MRSA persistence. And these sorts of individual studies often fail to capture the full mechanisms that are, powering MRSA persistence. And so our theory is that in order to better understand MRSA persistence, we likely need to integrate and consider all these modalities at once. And so in support of that, we've been working with some colleagues who have collected this large data set of 177 patients who were all admitted to a hospital with MRSA infections. And from each of these patients, they've derived up to 3 different types of data, including serum and plasma cytokine measurements, as well as gene expression measurements. And from each of these patients and from these different measurements we want to predict whether an individual's infection will persist after antibiotics, so this persister or APMB status, or whether it will resolve, so this resolver or ARMB status. The challenge, of course, working with multiple data types, though, is finding ways to properly integrate them. What we really want to do is to find these patterns of expression that exist across all these different data sources. And so we need some method to integrate and to factorize these different data types into something that later prediction models could actually use. And so here in the Meyer lab, the tool that we use is this process called Tensor Factorization, which is this data reduction technique that specializes in this really high dimensional representations of data. In many ways, you might view it as something like a higher form of PCA. So how Tensor Factorization works is we can represent our data into something like this, a cube, with these different modalities represented across these different axes. And Tensor Factorization, in turn, will take this cube of data and reduce it down to this series of components that are able to explain variance that we see across all these different modalities. Now, like PCA and other dimensionality reduction techniques, this is really powerful for helping to reduce things like noise. It's great for reducing data down. In some cases, it can be used to impute some missing values. And it's also, since these principal components are explaining variance across these different sources, we suspect that they might be able to capture mechanisms that are relevant across different modalities. And so as a quick comparison, we did mention the tensor factorization is similar to PCA, but part of the reason why tensor factorization is more powerful than this is in this sort of higher dimensional data representation. So in PCA, we're limited usually to these two-dimensional matrix representations of data. So if we had two sources of data, like two different types of cytokine measurements, we would have to concatenate these together into one singular matrix. In tensor factorization, though, we can represent this as a higher dimensional tensor, a sort of cube of data, with the benefit of this being that this better recognizes the interconnectedness of these different modalities. So by structuring our data as this tensor, it allows tensor factorization to recognize that there are connections between these cytokines, between these measurements. And we theorize that this will help to produce a better factorization and better recognize patterns that exist across these different data modalities. And so here's what our setup looks like. We use a slight variant on tensor factorization, which is called coupled matrix and tensor factorization, that provides pretty much the same benefit as most tensor factorization techniques, but it also allows us to append in this matrix of gene modules. And so our setup looks something like this, where we'll have a tensor of cytokine species against our different subjects and the two sources, so serum and plasma. And we'll couple it with this matrix of our subjects against our gene expression data. And from here, we'll use CMTF to factorize this coupled matrix and tensor into this series of components that are able to jointly explain variance across all of these different modalities. With the theory being that since these components are integrating these different sources, they might be able to recognize patterns and mechanisms that manifest across all these different modalities. And overall, we are finding that CMTF is really effective at reducing down our data. So over here on the left, we have this R2X plot depicting the amount of variance in our data that these CMTF processes are able to explain against the number of components. And we find that we're able to capture around 75% of the variance across all three of those modalities with just 8 components. The other thing to mention here is that, like PCA, CMTF looks to explain or to find components that explain the most amount of variance across the different data sources. And so something that we can do as a way to emphasize a certain type of variance or a certain type of data in CMTF is to scale up one of these different sources, like our cytokine or RNA, prior to factorization. And what we find is that increasing the emphasis of cytokine data just slightly tends to improve the accuracy or the ability of our model to factorize and to deconstruct our data, though having this RNA influence also still offers some benefit too. And what we also find is that these components are more predictive of persistence than these raw data types alone. So what we've done here is we paired a logistic regression model to these CMTF components to predict persistence. And as a baseline of comparison, we've also trained logistic regression models to predict persistence from these raw data types alone, like these plasma and serum cytokines, as well as these RNA modules. And overall, we're finding that CMTF universally outperforms these raw data models, especially if these plasma measurements are available. What's more, we also find that these components are not predictive of auxiliary clinical characteristics. So as part of our data sets, our colleagues also collected the sex, race, and age of each of our patients. And what we've done here is we've trained models to predict these different clinical characteristics from these CMTF components. And interestingly, we find that none of these models are predictive. So just as a quick note, in our data, there are 4 recorded races. So this 0.25 accuracy is exactly random chance. So overall, this is really telling us that our model isn't really predictive of these clinical characteristics, and that these components don't really capture variants relevant to that, which is intriguing for us in that MRSA persistence has been tied and has been correlated to things like sex and race before. So we are interested in that these components don't seem to be capturing any of that variance, and rather that they seem to be focusing more on mechanisms rather than variants that might be explaining these sorts of clinical characteristics that could also be influencing persistence. And one of the benefits of using this logistic regression model paired with CNTF is that it also helps us to quantify our associations between these different components in persistence outcome. So as part of the logistic regression model's training, it stores these coefficients associated with each of these different components that detail how that component is associated with persistence. And so what we've done here is we've run our model a whole bunch of times on bootstrap samples over our original training data. We've plotted out the different range of model coefficients for each of these different components. The thing to note about these scores is that both the sign and the magnitude are important. So components with a larger magnitude associate with a stronger predictive ability of persistence and are therefore likely more associated with persistence. And components with a positive coefficient are associated with the persister condition, whereas components with a negative coefficient are associated with this resolver condition. Overall, these analyses really highlight that components 3 and 7 demonstrate this strong association to the persister condition, while component 6 is our only component that associates with this resolver condition, though it has this really strong association and is the most influential of all of our components. And so with those component associations in mind, something that CNTF enables us to do is to backtrack through the factorization and identify what sorts of biological processes these components actually correspond to. So we originally structured our data as this tensor of cytokine measurements and RNA measurements. And how CNTF works is it looks to define these subject factors matrices and these cytokine source, and RNA factor matrices, with the idea being that it looks to try to reconstruct these original measurements as the product of the subject factor matrices with these other factor matrices. And so we can crack open each of these individual factor matrices as a way to evaluate how these components associate to these original signals, things like these different cytokines and RNA modules that we've collected. And so we can develop these really nice factor plots that plot out these components against each of the original cytokines and gene modules in our data as a way to sort of evaluate what sorts of signals and what sort of biological relevance might be behind each of these different components. And so from here on out, we'll start to interpret some of these components and figure out their biological significance. So the first of these that we want to talk about are components 1, 2, 4, and 5 that we've grouped together. On some of those previous plots, we showed that none of these components are all that predictive of persistence. And when we start to examine these factor matrices, we get a better idea why. In particular, we see that these have these really strong associations to batch effects. So when we plot out these component associations to our subjects, we find that the magnitude varies greatly depending on which cohort of patients it comes from. So we have this dashed line down the middle representing the 2 different cohorts, and we can see that the values for cohort 3 generally have a much lower magnitude than those for cohort 1, indicating that these components are probably capturing some variance relevant to this different cohort to cohort differences. When we break apart the cytokine signals, though, we do see some interesting biological signal. So in particular, we find these cytokines like IL-4, IL-5, and IL-13 all have these different levels of modulation across these different components. These cytokines themselves are usually associated with things like type 2 T helper cells, which we have found to be generally associated with MRSA infections, but they don't really offer much in the way of protection. And so we theorize that these components might be correlating to these Th2 responses that generally aren't that influential in MRSA persistence. Component 3 is the first of these components that has this association with persistence. As we show here, it has this association with the persister condition. And when we look at some of its cytokine signals, we see that it involves this strong downregulation of IL-4 and this strong upregulation of IL-12P40. From the cytokine angle, we suspect this component has implications with B cells. So IL-4 itself is used generally as a signal to help mature B cells. And IL-12P40 is the regulatory counterpart of IL-12P70, which has these sorts of documented crosstalk or mechanisms with B cells and these inflamed tissues. So overall, we're suspecting that from a cytokine angle, this is identifying this process that's suppressing B cells and their sorts of communications inside these infected and inflamed tissues. Now, what's more, this is further supported by the genomic signals or the transcriptomic signals we find from this component as well. In particular, one of the interesting modules we find for component 3 is this downregulation of P1 I11 M18, which through enrichment analysis shows these gene sets associated with things like neutrophil activation, which is telling us that this module or that this component is also downregulating this neutrophil activation. And given that neutrophils and B cells and all these different IL-12P70s have these sorts of crosstalk communication inside these inflamed tissues, that this component seems to really be suppressing this neutrophil activation and these sorts of communications that might occur inside these inflamed or infected tissues. Component 7 is our other persister-associated component. This one also demonstrates this strong association with the persister condition. And when we look at its cytokine signals, we see some interesting trends, and it involves the upregulation of IL-17A and GCSF. These are sort of interesting counterintuitive signals, and that both of these cytokines are generally associated with improved neutrophil production, which isn't something that you would normally think would be favorable for MRSA persistence. We find, though, that when we look at these genomic signals, that we kind of get a better understanding of how this might be pathogenic. Specifically, in looking at the gene module signatures here, one of the more interesting ones we find is this module P2I6M3, that when we do these enrichment analyses on, find that it has this negative regulation of erythrocyte differentiation, which is suggesting to us that this component also involves the prevention of the maturation and differentiation of neutrophils. So we theorize that this component might be identifying this process where it's improving neutrophil production, but keeping them naive and immature and preventing them from maturing into something that could actually clear out the infection itself. And then finally, component 6 are one resolver-associated components. In terms of its cytokine signals, we see some interesting overlap with component 7. In particular, we see this upregulation of GCSF again telling us that there's probably some improvement in neutrophil production. But interestingly, we also find this upregulation of growth, which is this really important cytokine in recruiting neutrophils to tissues and to also promoting their maturation. So we theorize that these two cytokines together give us this idea that this component is both improving neutrophil production and also ensuring that they're recruited and matured properly as well. In terms of other signals that we find interesting here, we find this downregulation of IL-17A, which we found to be upregulated in component 7. And we also see this downregulation of IL-10, which is this well-known immunosuppressive cytokine that MRSA has been documented to use as a way to prevent the immune system from responding to the infection. And so we theorize that this also is identifying this process where this component's preventing the suppression of the immune system from MRSA. Genomically speaking, we see a lot of similarities between components 6 and 7, but one of the more interesting ones that we find in one of the gene modules that shows a lot of difference between these two components is this P1 I11 M15 module, which is much higher in component 6 than it is in component 7. And in particular, we find that this module enriches significantly for this hematopoietic stem and progenitor cells, which is supporting that idea that we had further, where this component is involving both neutrophil production and maturation, since we're seeing these progenitor and stem cells being upregulated. Finally, as part of this model, we also wanted to evaluate if some of those individual CMTF components, or if a subset of those CMTF components might be as predictive as our existing model. And so we trained models using subsets of those components that we found to be important to persistence earlier, components 3, 6, and 7. And we found that if we looked at just components 6 and 7 individually, that we can see similar predictive accuracy to the full model altogether. And so when we plot out these different subject values for these components, we can start to see some interesting trends and some suggestion that there's probably a very strong heterogeneity in how MRSA becomes persistent. So when we plot out these component spaces for components 6 and 7, we can see this really clear cluster of patients in this top left that very strongly identify as this persister condition. But when we look towards the middle, we also see there's a lot of patients are persisters that don't have these same sorts of component associations. And what this tells us is that there's probably more mechanisms of MRSA persistence, and that there is some heterogeneity in which these infections can become persistent. So that each person's MRSA and how they become persistent can be different. So yeah, so just a quick set of conclusions. We find that CMTF effectively reduces and integrates biological modalities, and this integration leads to components that are more predictive of MRSA persistence than these sorts of singular data types alone. We also find that these component interpretations are really beneficial and that they offer these unique insights into these integrated persistence determinants. For component 3, we found this link between the suppression of neutrophil activation and B cell crosstalk to this persister condition. And for components 6 and 7, we identified that both erythrocyte production and differentiation are key determinants of MRSA persistence, and that you need both in order to favor that sort of resolving MRSA infection. In terms of some quick next steps, one of the things that we're really interested in exploring with this tensor factorization scheme is multi-infection models. So as we showed earlier, you can do these sorts of couplings between tensors and matrices as a way to increase the amount of data that you can share across them. And so here we're proposing this sort of ambitious scheme where we also want to couple in another type of infection. So up here at the top is our MRSA data set that we've been talking about in this presentation so far. And then here at the bottom, we want to introduce another data set for Candida albicans, which is this fungal infection that shares a lot of similarities with MRSA, and that it tends to be a community-acquired infection that primarily favors immunosuppressive or patients with immunodeficiencies. And so we theorize that this additional coupling could help in a few ways, and that it could add more data for us and help our tensor factorization methods better understand the sorts of connections between these different modalities. So understanding how these different cytokines and gene modules might relate. And it could also help us to better understand each of these infections individually, as well as sort of identify generalized immune weaknesses and shared pathways of immune evasion that exist across them. Really interestingly, we find that both MRSA and Candida albicans have some similarities in how they evade the immune system. And so we think this coupling could really help us to better understand each of these infections individually. So that's just a quick set of acknowledgements. Thanks, of course, to the entire Meyer lab. In particular, thank you to Mark Krexel over here with his dog, Leo, for all of his great help in working with gene enrichment analyses. Thanks to Cyrilus Tan over here on the far left for all of his great help with tensor factorization. And thank you to Scott Taylor, our lab manager, for all of his assistance in getting me started with MRSA and with teaching me a little bit more about the data set. Thanks, of course, to the MRSA Systems Immunobiology group, and thank you to my dog Kiki for being a pretty good coworker throughout this entire pandemic. So thank you all for listening, and I'm happy to take any questions at this time.